Life, in all its magnificent diversity, is a story written in the language of inheritance. From the towering redwoods to the humble earthworms, every living organism carries within it a blueprint that dictates its form, function, and destiny. This blueprint, passed down from generation to generation, is encoded within a molecule called deoxyribonucleic acid, or DNA. Understanding the molecular basis of inheritance is akin to deciphering the very essence of life itself. It unravels the mysteries of how traits are passed from parents to offspring. It allows us to comprehend the intricate mechanisms that govern the diversity and evolution of life on Earth. Join me on this extraordinary journey as we delve into the microscopic world of genes, chromosomes, and the molecules that hold the key to our existence. Our exploration begins with the molecule of heredity itself, DNA. Imagine a vast library filled with countless volumes of information. Within these volumes lie the instructions for building and maintaining a living organism. DNA is the language in which these instructions are written, and genes are the individual words that make up this intricate code of life. This remarkable molecule, DNA, holds the key to understanding who we are and where we come from. It is the thread that connects us to our ancestors and the bridge to the future generations that will inherit our planet. In the heart of every cell, within a tightly guarded vault called the nucleus, resides the molecule of life DNA. This remarkable molecule, often described as the blueprint of life, carries the genetic instructions that dictate everything about an organism, from its physical appearance to its internal workings. But what exactly is DNA, and how does it store such a vast amount of information? To understand the secrets of DNA, we must journey into the realm of the very small, to the level of molecules and atoms. DNA, it turns out, is a long ladder-like molecule twisted into a graceful spiral known as a double helix. Picture a rope ladder twisted upon itself, and you have a good visual representation of DNA's elegant structure. The sides of this ladder are formed by alternating sugar and phosphate molecules. Attached to each sugar molecule is one of 4 nitrogenous bases: adenine, A, thymine, T, guanine, G, and cytosine, C. These bases, like letters in a genetic alphabet, hold the key to life's code. The true beauty of DNA structure lies in its elegant simplicity. The 2 strands of the helix are held together by hydrogen bonds, weak chemical attractions that form between specific base pairs. Adenine always pairs with thymine, AT, and guanine always pairs with cytosine, GC. This predictable base pairing, known as complementary base pairing, is crucial for DNA replication and the faithful transmission of genetic information. Life is a continuous cycle of growth, renewal, and perpetuation. At the heart of this remarkable cycle lies the ability of cells to divide and create exact copies of themselves. And at the heart of this cellular reproduction lies the elegant process of DNA replication, where the master blueprint of life is faithfully duplicated. Imagine a cell preparing to divide, like a bustling city about to create a perfect replica of itself. Before the division can occur, the cell must first make a copy of its precious library of genetic information, its DNA. DNA replication is a marvel of molecular choreography, a carefully orchestrated dance of enzymes and proteins that ensures the accurate duplication of the DNA molecule. The process begins with the unwinding of the DNA double helix, like the slow separation of a zipper. As the 2 strands separate, each strand serves as a template for the synthesis of a new complementary strand. Enzymes called DNA polymerases play a crucial role in this process. They move along each separated strand, reading the sequence of bases and adding complementary nucleotides to the growing new strand. Adenine, A, on the template strand will attract a thymine, T, on the new strand, and guanine, G, will attract a cytosine, C. This precise base pairing ensures that the genetic information is faithfully copied. The result of DNA replication is 2 identical DNA molecules, each consisting of 1 original strand and 1 newly synthesized strand. This is known as semi-conservative replication, a term that reflects the fact that each new DNA molecule conserves 1/2 of the original molecule. Section 4. Transcription. From DNA to RNA, a message unveiled. The DNA molecule, safely housed within the nucleus of a cell, holds the master blueprint of life. But how does this genetic information, encoded within the sequence of DNA bases, get translated into the proteins that carry out the myriad functions of life? The answer lies in a fascinating process called transcription, a crucial step in the journey from gene to protein. Imagine a scribe meticulously copying a page from a precious ancient text. In much the same way, transcription involves creating a messenger molecule that carries the genetic instructions from the DNA in the nucleus to the protein synthesis machinery in the cytoplasm, the bustling factory floor of the cell. This messenger molecule is called ribonucleic acid, or RNA. Transcription begins with the unwinding of a specific section of the DNA double helix, exposing the gene that needs to be transcribed. An enzyme called RNA polymerase then binds to the DNA strand at a specific site called the promoter region, marking the starting point for transcription. RNA polymerase then moves along the DNA template strand, reading the sequence of bases and synthesizing a complementary RNA molecule. The process is similar to DNA replication, but with one crucial difference: instead of thymine or T, RNA uses uracil or U as a base to pair with adenine or A. The newly synthesized RNA molecule, known as messenger RNA or mRNA, carries the genetic instructions from the DNA in the nucleus to the ribosomes in the cytoplasm. The mRNA molecule is like a blueprint, a temporary copy of a gene's instructions, ready to be translated into the language of proteins. Section 5. Translation. Deciphering the genetic code into proteins. In the bustling factory floor of the cytoplasm, the genetic message carried by messenger RNA, or mRNA, is translated into the language of proteins. This intricate process, known as translation, is the final step in the journey from gene to protein, where the genetic information encoded in DNA is ultimately expressed as a functional molecule. Imagine a bustling assembly line where skilled workers decipher a coded message and use it to build a complex machine. In the cell, ribosomes act as these skilled workers, reading the mRNA code and using it to assemble amino acids, the building blocks of proteins, into long chains called polypeptides. The mRNA molecule, carrying its genetic message, attaches to a ribosome, the protein synthesis machinery of the cell. The ribosome then moves along the mRNA, reading the sequence of bases in groups of 3 called codons. Each codon specifies a particular amino acid, the building block of proteins. As the ribosome reads each codon, it recruits a molecule called transfer RNA, or tRNA, which carries the corresponding amino acid. The tRNA molecule, like a delivery truck carrying a specific part, brings the correct amino acid to the ribosome, where it is added to the growing polypeptide chain. This process continues, codon by codon, amino acid by amino acid, until the entire protein molecule is synthesized. The polypeptide chain then folds into a specific 3-dimensional shape, determined by the sequence of amino acids, and the protein is ready to carry out its designated function.